and it's great to talk to you guys today about the research that we've done in our lab. And feel free to ask questions if you have any. Okay. So today I'm going to introduce you two topics. One is relates to forward programming of stem cells. Another one is related to genome editing. So first of all, the stem cell research, if we consider the pluripotent stem cells, that is back to 1998 when James Thompson derived the human embryonic stem cells. So it has been more than 20 years now. And until now, you can see the map here. There are many clinical trials that going on now, and that is focused on many different type of diseases, like in the brain, like in the heart, the immune related, and endocrine that relates to the diabetes. Yeah, you can see the cumulative trials over time. So now overall, we have more than 100 clinical trials going on until 2025. Okay, so it's a very, very exciting field. And if we count this clinical trials based on different countries, US is still the largest portion, that is 38%, and then that comes followed by China and Japan. So hopefully we will see more and more of these clinical trials become the real cell-based therapies, that are approved by FDA, and then we can use these to treat the disease. Okay, so one of the most important thing for using these pluripotent stem cells, which has been has human embryonic stem cells and induced pluripotent stem cells. So first of all, these cells cannot be used directly as a therapy. So they are very different from the adult stem cell, just like the blood stem cells. You can use them directly for transplantation. But for pluripotent stem cells, you cannot do that. If you transplant the pluripotent stem cells, you will introduce teratomas. Basically, these cells will form tumor-like tissues in vivo. So that is dangerous for the patients. So in order to use these cells, you have to differentiate the cells to become the cell type that you are interested for therapy. For example, for heart failure, we are interested in cardiomyocytes as a therapy candidate. For diabetes, we are interested in pancreatic islet cells. So the question and the most challenging question here is the differentiation. How do you convert these cells to become the cell type that you're interested in? Okay, this is a very, very difficult question. So I'm going to show you one of the example that I did before for converting these cells to become cardiac cells. Okay, the spontaneously beating cardiac cells. So these are the cardiomyocytes that generated from pluripotent stem cells. And we can also use these cardiac cells to engineer cardiac tissues. So you can see these tissues are linked, with these two ends that can be used to measure the force generated by this engineered cardiac tissue. So how do I do that? So basically, in order to achieve this differentiation, I use chemical and biological way to modulate this Wnt pathway. So it turns out that this Wnt pathway is a very critical one regulating stem cell differentiation. So basically, in a short word, what I did here is we are using one of the chemical to activate the Wnt pathway, and then after day 1 to day 5, we test repression of this Wnt pathway at different time points. So you can see at around 36 hours, if you inhibit the Wnt pathway, you can achieve the maximal cardiac cell production. Okay. So if you see this figure, we test inhibition of Wnt pathway every 3 hours, between 24 and 48 hours. Okay. So again, it shows like the 36 hours is the best. Okay. So you can see the timing is very, very sensitive. Cells need to sense the correct signal at the right timing. You can see if you inhibit the Wnt pathway at 24 hours or 48 hours, the cardiac production efficiency will be much lower. Okay. So this is also the reason why differentiation is so difficult, because you don't know when is the optimal time to treat the cells with the correct signal, right? So this is the paper we published about more than 10 years ago. And then, that is example of cardiac differentiation. So, that end up with a very simple protocol. You needed to use two chemicals, one to activate the Wnt pathway, and a few days later, you repress the Wnt pathway. But you can see some of other differentiations could be even more complicated. So this one we are talking about is the so-called pancreatic differentiation. So this differentiation protocol is very complicated. So you can see this is the first 4 days. And here is all the factors you need to add into the media, okay? And with their concentrations. And each of these concentrations are optimized. So for example, how do you know, this guy activin A needed this concentration? So it's a work of many, many labs, and they optimize for many, many, many years, okay? And then you can see for day 5 to day 6, and then you need to add these factors, okay? And day 7 to day 10, you needed to add these factors. So complicated, because a natural question to ask is, how do we know this, right? Indeed, nobody knows. Long time ago, many, many labs spend like 15 years and work this out, okay? So you may think, can I get pancreatic cells after day 10? The answer is no. You need another like 2 weeks, only until like about day 25 and day 30. And then you need to do these things again, especially you can see this stage. This is so complicated. And then each of them have its concentration. Think about it. How can you figure out this, right? So that's why, the traditional direct differentiation for some of the cell types, it's very, very difficult to develop, and it costs numerous funding and many, many years, okay? So because of this, I started to think, can we have a more straightforward way to generate the desired cell types from stem cells instead of using the growth factors, chemicals, and modulate at different stage, okay? So the way that we can do stem cell differentiation, the traditional one is called direct differentiation, right? You are using, growth factors or chemicals, and then you add at different stage. So this method is very, very complicated. So let's say we have a very simple differentiation, only takes about 7 days. And then you have, you read the literature and you think that you may need 4 growth factors, and they are using different concentrations, right? And then if you want to fully optimize all of them, because maybe growth factor A, you need to add here, add here, add here, or add here. And then at different time points, they may require different concentration. So growth factor A, maybe here is 10, here is 0.5, here maybe 50. So if you test all these combinations, guess how many combinations we have? You will have a combination number like this. So this number is beyond the capability of all the stem cell labs in all around the world. And if we test all together, we cannot do this, because this is just like there's no way you can test this number of experiments, okay? So you can see even this simple example, if you want to fully test all the combinations, that is impossible. Not to mention, if you have a differentiation, takes 30 days, that's impossible. Okay. So another way that you do it is the so-called forward programming way. So basically, you start with stem cells and then you want to generate a cell that you are interested in. Let's say you want to generate pancreatic beta cells. So this approach is we are not going to figure out what growth factors we need. Instead, we are going to figure out what are the key transcription factors that is important for beta cell. And then we put these TFs directly into stem cells and hope the stem cells can gradually become beta cell. Okay, so this is more like a gene approach. So there are two pioneer papers in the field. One is from the Church lab, one is from the Dr. Feng Zhang lab. And they have established the human TF library. So basically, all the genes encoding human TFs are available, okay? You can get all the genes. And then in total, we have about 1,500 human TF genes. And from these 1,500, there must be a good combination that you can put them into stem cells and you can generate beta cells, okay? Or if you want to generate some immune cells like T cells, you can do the same thing, okay? But the question is, again, it's also not trivial. It's also not easy because how do I figure out, which genes, which TF genes that I should do it, right? That's also very challenging, okay? So in the Church lab, what they did is they first tried to see among these 1,500 genes, which single TF can induce differentiation. So what they did is they encoded these genes into different viral particles and then use these viral particles to infect human pluripotent stem cells. And then they wait about 4 days. After 4 days, they started to, sort out the cells based on the TRA160 expression. So if the cells still express this marker, they are still stem cells. But if they lose expression, that means they differentiated. So they will check these cells and see which TF is in this cell. Okay. So based on this assay, they find that there are about 100 genes by themselves can cause differentiation. And they have a few examples. So one of the examples is called ETV2 gene. So this gene by itself, when you put this gene into cells, into stem cells will become endothelial cells, which make up of our blood vessel. And then another gene is called 3.1, NKX 3.1. When you put this gene into stem cells will become neurons, okay? And there's another gene called ATOH1, and the stem cell become fibroblast. So good. But this is only testing like the single gene. If you do this, basically if you work really hard, you can do it because in total you will only have 1,500 experiments to do. That's still a lot, but it's doable, right? If 10 labs work on this together, each lab do 150 genes. Yeah, that's doable. You still can do it. But what about if you want to test two genes combination out of like 1,500? Then that's more challenging because you are basically like doing the combination that is pick 2 out of 1,500. You guys know math, you are from engineering, so you can calculate how many combination this is, okay? Not to mention, what if you want to test 3, right? Pick 3 out of 1,500, then you got even more, okay? That is very, very challenging, okay? But again, this is not easy, but the direct differentiation is also can be even more difficult, okay? So what my lab did is for this forward programming is, we generate a drug inducible system. So basically, you can clone whatever gene you're interested in here. And then from this construct, if you use a drug called doxycycline, dox, and it will induce expression of the gene that you clone here. Okay. Here is showing the dynamics of the gene expression. So there's time 0, you add the drug doxycycline, you can see gradually cells turn on the expression. And once you remove the drug doxycycline, the expression is going down. Okay, so it's a inducible and a reversible expression system. So from this, I started to work on endothelial progenitors, see if we can find any other factors that can do this job. So remember previously, scientists already discovered ETV2 can do this, right? So my question is, can I find another factor which is different from ETV2, but it can also do this? So in order to find that, I need to find the genes that enriched in the endothelial cell progenitors, okay? So what I did is do the differentiation, and then on day 5, we did the single cell RNA sequencing. And it turns out we have a differentiated population here, and this population enriched for 3 endothelial gene, V cadherin, CD34, CD31, okay? And then based on this, and we find 3 transcription factors are also enriched in this population. Okay, you can see only this population. So these genes are called SOX7, 17, and 18. And then we decided to do immunostaining and check. Indeed, we find this SOX17 gene, is co-localized with the endothelial marker V cadherin. And then next, we decided to see whether this gene plays critical role during endothelial cell differentiation. So what we did here is we decided to, overexpress these SOX genes and then see if we can have enhanced differentiation. Indeed, what we find here is, if you do the SOX17, you got like a higher expression as compared to the black one as a control, okay? And then when we do the Western blot, we find the SOX17 gene indeed turn on at day 3, day 4, day 5 differentiation, which is around a similar time like the endothelial marker V cadherin. They are also turned on, at this time, okay? And then if we quantify the percentage of SOX17 cells, and we find the cells express SOX17 gene unknown, and then later, they become double positive, okay? So this indicates SOX17 may play a critical role during this differentiation. And next, we did the lockdown experiment to see if this gene is indeed important. So if this gene is important, if we get rid of this gene, it should inhibit the differentiation. And indeed, we find that. So you can see here, the differentiation efficiency become lower, when you add DOX to repress the SOX17 expression, okay? So next is the most exciting part. We try to understand whether SOX17 unknown, overexpressed SOX17 unknown, is sufficient to program stem cells to become endothelial cells. So previously, scientists already discovered ETV2 can do that, right? So we use ETV2 overexpression as the control. So you can see, indeed, ETV2 can overexpression can induce the endothelial cell marker expression. That means program stem cells to become endothelial cells. And encouragingly, SOX17 can also do similar job. Okay, SOX17 can also do similar job. And you can see the expression of CD34. When you overexpress SOX17, starting on day 1, immediately cells started to express CD34. Okay, so it's very, very rapid. And then we test the duration of the SOX17 expression. And turns out, we need at least about like 4 days to program cells to endothelial cells with SOC17. Okay. So, and you can see here, we also test the doxycycline concentration because this controls the amount of SOC17 expression, right? So it shows that you need a 500 nanogram per ml in order to achieve the forward programming, okay? And the efficiency we got here is about 43%. And next, we optimize the stem cell culture conditions, either use mTESR or E8. You can see if you use E8 to culture the cells, you can further enhance the forward programming efficiency by SOC17 to 92%. Okay, this efficiency is very high as compared to what we achieve here. Here is like we only have like 15. Okay, so we achieve the efficiency from 15 to 92%. So basically, you got relatively pure endothelial cells. And from these endothelial cells, and we show these cells are functional. So when you put these cells on top of matrigel, they can form tuber-like structures, and they can also uptake the acetyl LDL, just like the primary endothelial cells will do, okay? And they are also response to the inflammation factors, such as the TNF-alpha. So basically, overall, we revealed a novel forward program approach to make endothelial progenitors and endothelial cells, which is using the gene called SOC17, okay? Next, I'm going to change the gears a little bit. I'm going to talk about the genome editing of stem cells, okay? So why we care about genome editing in stem cells? The reason is when you use genome editing, you can create lineage-specific reporters, okay? So for some type of cells, such as cardiomyocytes, if you do not have reporters, you may be fine because you can identify the cells based on, they are spontaneously contracting, right? Under the microscope, if you see they are spontaneously contracting, you got the beta cell, cardiomyocytes. But for some other cells, like beta cells, pancreatic beta cells, just based on the morphology, you cannot tell, if you got beta cells or not. Okay, so in that case, you have to use some of the reporters to help you understand if you got beta cells or not. So you can put a green fluorescent protein under the control of beta cell specific gene. So in this way, when the cells become beta cell, they will become green fluorescence under the fluorescent microscope. You can identify them, right? And the gene editing can also be used to generate lockout cells. And why we care about this? So using this, we can study biology. So for example, if you are interested in whether the Wnt pathway is important for beta cell differentiation, you can lock out the Wnt pathway key gene like a beta-catenin, and then see if you can use the lockout cell to make a beta cell. If you can still do it and very efficiently, that means the Wnt pathway is not needed for beta cell differentiation. If you cannot do it, then that means the gene you lock out, like beta-catenin, is required. Okay, so you can use this to study the gene function for all human genes. Okay, and another thing is, sometimes you get the patient cell and they have their mutations. You can use gene editing to correct the mutation. Okay. So first, let's introduce the reporter, okay? So why we are interested in this? So for example, my lab published a paper say we can use modulation of the Wnt pathway to make the anadrome cells. And one of the marker here is SOC17. So the review asked, how do you know that you can do this and get a SOC17? So I decided to use the TT tomato lock-in cell line. So basically, TT tomato is controlled by SOC17. And this video is a 2-day video. Okay, so every 20 minutes we take a picture and then we make a video. So you can see at the very beginning, it's dark. And then gradually SOC17 started to express. When SOC17 express, they will be labeled with TT tomato. So in this way, you can conclusively say that, during the differentiation, my cell become SOC17 positive endotherm cells. Okay. All right. So, but the traditional locking methods suffers from many aspects. The first one is time consuming. You can see here, this is the traditional lock-in. So basically you have some donor DNA, you have some Cas, and then you deliver them into stem cells. And because the efficiency is low, so you need to use a drug selection cassette. So you use the drug to select the cells, that has these constructs, okay? So, and then in order to identify, which cell has the lock-in, for example, SOC17, right? As I showed you before, the undifferentiated cells does not show fluorescence. So then you have no idea which cell has the lock-in, which one is not, because the successful lock-in and the one that is not successful lock-in, both of them are not fluorescent. So you don't know which one is the correct one. So what you can do is single cell replating them and wait until they become colonies and take their genome DNA and then do PCR and then try to see which one has the TD tomato lock-in, okay? And then you need to do one more round. Why you need to do that? Because you have the drug selection cassette. You need to do one more round and use Cree to remove the drug selection cassette. So that's why overall it took about 1 month. And many times it can be more than 1 month. And the lock-in efficiency is so low. So sometimes, let's say, if the efficiency is 0.1%, that means among these colonies, only 0.1% of them are the correct lock-in. So then think about it. If the efficiency is this, and then you need to do at least 1,000 PCR, and then you can find one that is the correct one. Think about it. Can you do 1,000 PCR? Just pipetting, keep pipetting, and then do 1,000 PCR. This work is become infeasible or impossible for many, many labs. I can hardly think of anyone can do this. And if the efficiency is 0.1%, you do 1,000 PCR, it doesn't mean you will be successful because of the statistics. You probably need to do 2,000 PCR in order to find one. So basically, this becomes impossible. So this is also the reason why we do not have that many lock-in reporters. It's just it's just too difficult to do, okay? So now I'm going to solve this problem by two approaches. The first one, the efficiency is low, right? So how do we improve the efficiency, okay? So we are going to improve the efficiency. And then the second problem is, you needed to do these genomic PCR in order to identify which one is the correct one. Can we then do it, but still isolate the lock-in clones? Can we develop a smart way, a clever way, a better way to do it, to get around it? Okay. So we are going to solve both problems. So in order to solve the first problem, instead of use the plasmid DNA for gene editing, we decided to use modified mRNA. Okay, so we use modified mRNA to encode Cas9, and then the advantage of modified RNA is it's robust and efficient, and it's non-integrating, and the expression is high but a short duration. So the Cas9 do not stay in cells for a long time. So in this way, it will reduce the risk of off-target activity, okay? So when you use a plasmid DNA to express Cas9, so for example here, there's a problem. In some cases, so you can see the clones here, in some cases, these plasmid DNA can somehow integrate into the human genome. You can see the sequencing data here. So this is bad because when you do the patient IPS mutation correction, you correct the patient mutation, but then you introduce another mutation in another place. Then what's the point, right, of doing it? So that's why using the plasmid DNA, have a big problem. Okay, so instead we don't want to do that. We do the modified mRNA. So the mRNA will not integrate into the genome. Okay. So if you check the mRNA transfection efficiency, you can see the efficiency is very, very high, around 90%. And with Cas9 modified mRNA, so now we are doing a gene knockout. So this gene is called CD90. It's naturally expressed at the stem cell stage. You can see if we do the modified mRNA with the enhancer molecule p53DD, our knockout efficiency is 84%. Instead, if you just do the plasmid DNA, the efficiency is like 14%. Okay, so it's a huge difference. So by doing this, we solve the problem of low lock-in efficiency. Now our efficiency is high. Okay, so the second one is still, your efficiency is high, but you still need to isolate the clones and then, do the PCR, right? And then that takes a long time, right? Can we get around it? That is part two. So we thought about this and we come up with a very clever methods. So we can isolate the lock-in clones without doing PCR. So how to do that? We are going to temporarily activate the unexpressed gene, the silence gene, such as SOC17, with a system called a model SAM. So what exactly is model SAM? So basically, we use a data Cas9. So this Cas9 will not be able to cut the DNA, but it can still bind into DNA. So we use this dCas9 activator and with its specific ggRNA. And for this, we are going to design the DNA binding sequence at the promoter region of the gene. So for example, if we do the lock-in of SOC17, and this is the promoter region, we will bind here, okay? So once this Cas9 activated binding here, it will not cut the DNA. Instead, it will have the activation domain by the P65 and HSF1. So which will recruit the RNA polymers and then turn on the downstream gene expression, okay? So if a cell has successful lock-in, by doing this, we will turn on the fluorescent gene expression, such as TD tomato or GFP. So in this way, we can distinguish the successful lock-in and unsuccessful lock-in, okay? So this is a smarter way of doing isolation of the lock-in cells, okay? And then based on this, we can also do the deep learning framework to identify where is the best binding location in order to activate the gene, okay? So for example, this is use the SOC17 as example. So this blue stuff here is the SOC17 gene. And then this region is the promoter region. So you can design many different potential guide RNA, and the deep learning training model can help you identify which one is the best in order to activate the SOC17 gene, okay? So let's test this idea to see if this works, okay? So first, we use a already established SOC17 reporter cell. So it's called a SOC17 TD tomato, okay? We just want to test if indeed we can activate this gene at the stem cell stage. So what we did here is we are going to transfect the cell with a dCas9 model RNA and with SOC17 guide RNA, okay? And then after, here is the time. So you can see 12 hours later, you already have a little bit TD tomato positive cells. And the maximum TT tomato positive cells happens at 36 hours, okay? And then the expression is going down. Why is that? Because we use a modified mRNA expression for for the DCAS9 activator. The modified mRNA expression only happens in the cell for 1 or 2 days. So once the DCAS9 is gone, the expression will go down. So you can see this expression is a temporary expression. It goes up and going down. So this is very good because it gives us a window. So if by this time, if we go to the fax sorting facility and we sort out our successful lock-in cells, and then later, they shut down expression of these fluorescent protein. That's exactly what we need. Okay, so based on this experiment, we identify the best time window for the isolation of the successful lock-in cells. So you can see we call this a model CM system for activation. Not only we can activate SOC17, we also activate many other genes, such as PDX1, NKX6.1, right? We can activate so many different genes. And it turns out, indeed, these activations are transient. And you can see after activation, 5 days later, cells become stem cell again. So this is activation for PDX1, SOC17, and NKX6.1. You can see 5 days later, the cells still express prepotency markers. They are still stem cells because 5 days later, they will stop the expression of these differentiation gene, okay? So based on this, we develop our magic methods. So it stands for modified RNA based activation for gene insertion and lock-in. So 2-step. Step 1, you do the gene lock-in, okay? So basically you do the Cas9 modified mRNA, and then with a mini donor, because our efficiency is so good, we do not need the drug selection cassette, okay? Because we do not need the drug selection cassette, and then later you do not need to remove it because you didn't have it. And that's step 1. And then within this mixture, you have successful lock-in cells. Let's say this guy is a successful lock-in cells, okay? But the bad news is you based on your eye, you don't know which one is a successful lock-in cells. That's why we have our step 2, the gene activation. It's a very smart approach. So we do the DCAS9 activator with dgRNA, and we activate it. So now you can see you have some cells, this cell. Now you turn it on, and then you can do fax sorting. You can isolate it, okay? So 2-step. And here we compared if you use modified mRNA versus plasmid DNA to do the magic approach. It turns out the modified mRNA approach achieve much higher efficiency as compared to if you use plasmid DNA. Yeah, this efficiency is very, very low. It's only 0.08. But if you do modified mRNA, it's much higher. That's because when you do the lock-in, for here, if you do the modified mRNA, the efficiency is much higher than the top. Use the DNA, okay? So now we have developed a completely new methods. Now we can use these methods to make gene lock-in for whatever gene you are interested in, okay? So let me show you a few examples. So NKX2.5 is a cardiomyocyte gene. And then you can see here, we can label this cardiomyocyte gene with the GFP. So you can see our efficiency is about 6%. And then based on this, we do the sorting, and we got the GFP positive cells, and indeed they express NKX2.5, okay? And then we do the differentiation with our reporter cell, and indeed, on day 10, we see the GFP positive cells. And then when we do the staining, you can see indeed the GFP cells labels with the NKX2.5, okay? And then when we, basically from here, we already got the lock-in reporter cells. So there's no need for us to do single cell cloning, but we still do it. The reason is just for publication purposes, just to show, these sorted cells are indeed, you can see clone 1 to clone 12. You can see indeed they are 100% are lock-in clones, okay? And then we do another gene called the PDX1 gene. So this gene is a beta cell, pancreatic beta cell specific gene. And this gene, the efficiency is a little bit lower. It's like 0.44. But the good news is even the efficiency is 0.44, we can still do the FACS sorting and then to get the correct lock-in cells, okay? So you can see here, after sorting, we have the green cells and they are PDX1 positive, right? And then after sorting, if you culture for another 5 to 10 days, cells still behave like undifferentiated stem cells. And then using these reporter cells, you can do differentiation. So you can see this is the stage 4 of the beta cell differentiation. You can see cells turn on GFP expression when cells started to express PDX1, okay? So you can see this is on day 14. Cells are green, cells also express PDX1, okay? They are double positive. And then, we also do example of SOC17 lock-in, okay? Although we already have a SOC17 reporter cell line, we decided to make the more SOC17 lock-in reporter cell lines in, other stem cell lines. And it turns out we can also do SOC17, and the efficiency is about 0.5%. And you can sort the cells out, and then you can, use these cells as the reporter cells, okay? So you can see, use these sorted reporter cells, and then we do the endoderm differentiation. You can see on day 4, some of the cells become green. And indeed, it reports the expression of the SOC17. So the green cells are SOC17 positive cells, okay? And then based on this, you will see, all the clones are the correct lock-in clones. Basically, the efficiency is almost 100%, okay? So this is our magic methods for do gene lock-in. So in summarize, you need about 1 week to do it. Here, you need about 1 month, okay? And greatly shorten the time. And here is just 2 steps. You don't need to do PCR, okay? There's no PCR at all. So step 1, you are going to use Cas9 donor DNA and deliver into stem cells. And you wait for 2 to 3 days. And then on day 3, you are going to transfect cells with DCAS9 activator and the dgRNA to activate this gene. And then you wait for another 2 days, and then you sort out the cells. That's it. So in total, you have about like 5 to 6 days. Yeah, less than 1 week. And you got your reporter cells, which is much, much easier than the traditional lock-in methods, okay? And also, if your efficiency is low here, let's say 0.2%, 0.1%, you can still do it. All you need to do is you just transfect more of these cells and then just do sorting, okay? You can still do it. But here, if you got the efficiency is 0.1%, as I told you, there's almost no way you can do it, okay? Because you need to screen out more than 1,000 clones. Think about it. Do you have like incubators that you can put 1,000 dishes into your incubator? And then from 1,000 dishes, you are going to isolate the genomic DNA for 1,000, tubes. Just think about 1,000 tubes you put on your bench. You probably even label from 1 to 1,000, it already took you like 1 hour. You know what I mean? And then not to mention, you are going to isolate the genomic DNA and do PCR. Seems like nobody can do it. Not to mention, what if your efficiency is 0.01%? Then forget about it, okay? Then here, you may still do it as long as you just activate more cells, right? You have more dishes of cells, and then you can still sort as long as you have more input cells here. You sorted the cells. Even you have like a few thousand of your sorted cells. Let's say you got 2,000 of sorted cells or 1,000. Once they attach to the dishes, you can still expand them and culture them. Okay, you can still got your locked-in cells.